Hey you guys, welcome to chapter 17.5 notes. We are at the last section of the chapter 17 and that is no small feat. So nice job, keep up the great work, and let's be done with this very soon. So here we are, mutations. Earlier in the chapter we talked about large-scale mutations that occur during exon shuffling. So let's talk about some smaller mutations now. For example, point mutations. A point mutation is going to change one nucleotide in the genetic sequence, and that's going to cause potentially a change in the amino acid sequence. So if mutations occur in a gamete, so in the sperm or the eggs, it can be passed on to the next generation. However, if you get too much sun on your arms and you have skin cancer, that is not a mutation that you're going to pass on to an offspring. So generally, mutations they can be advantageous. They can lead to adaptations, but they generally are detrimental or silent. If it does have an adverse effect, then it is considered to be a hereditary disease or a genetic disorder. An example of this is sickle cell anemia, and it's caused by a single base pair. So right here we can see one single point mutation. So the original genetic sequence is CTC. However, it became CAC. So that is going to, instead of coding for a glucosamine, it's going to code for a valine. And that is going to cause the red blood cell to have this sickle shape. And that is going to cause a lot of negative consequences. So that is a point mutation. And so when mutations occur in portions of the DNA that are not coded in the proteins, so if it's not expressed, then it'll have no effect on the phenotype. Small-scale mutations that occur within the protein coding portions of a gene can be broken up into two sections. So we're going to talk about single nucleotide pair substitutions and nucleotide pair insertions or deletions. These are the ones that have larger effects. So let's start with the single nucleotide pair substitutions. And this is very simply if we just when one nucleotide is replaced. So if we look at the genetic code here, the original strand, it was CCG ATT, and we've got a CCA ATT, so the A replaced that G. That is going to be a point mutation. However, if you notice, both CCA and CCG both code for glycine. And so therefore we call that a silent mutation because the same protein will still be translated. If the substitution is expressed and changes an amino acid, then we call it a missense mutation. So for example, let's go back to our sequence here. It was CCG ATT. Now it's TCG ATT, so a T instead of a C, and that changed the amino acid to serine. And serine is a different amino acid, and that's going to change the sequence of amino acids, therefore the shape, and therefore the structure. If a substitution occurs, and instead of coding for a different amino acid, it creates a stop codon. So remember that there are no amino acids for stop codons. And so I like to remember this as, it's called a nonsense. And so non or none reminds me that there is no amino acid added. So nonsense or nonsense or none amino acid. And if that's going to cause a stop codon, which means that it's going to, the amino acid sequence will be released and the protein will be cut short. All right. Insertions and deletions, or additional or lack of nucleotides, they're going to have a large impact. This is called a frame shift. If it happens in a non-coding section of a gene, it is not referred to as a frame shift. However, it still can impact the phenotype. And so what happens here, let's go back to our original sequence. We had CCG-ATT, or let's start before that, AAA, CCG. We have AA, oh, we're missing an A, and so it goes CCG. So that changes not only this leucine, but everything else after it is going to be different. So that is the whole frame is shifted over, and so that's going to really have cause a lot of missense. It's going to have a very negative impact. That protein is pretty much garbage after that. So mutations can occur during replication. So if it's replicated incorrectly, during recombination. So if a protein during, let's say, crossing over is recombined and it causes a mutation, that can happen during transcription and or translation. However, if a mutation is on the DNA, it's going to be expressed over and over again. So this is a bigger problem. Whereas if it's a mutation during transcription or translation, it will only be expressed one time in one protein and really is probably pretty negligible. And so when mutations occur during DNA, RNA, protein, or during the central dogma, and it is not fixed by proofreading enzymes, we call this a spontaneous mutation. It's spontaneous as opposed to induced by a mutagen, or an induced mutation by a mutagen. So it's spontaneous. It just happened during replication or something, and it just wasn't fixed. So it was spontaneous. It wasn't caused by something outside in the world. Now, let's talk about mutagens. These are things from the outside world that are actually going to impact the genetic material. So if we see here, we can have nucleotide analogs. These are chemicals that are actually similar to nucleotides and can take the place and incorrectly pair during replication. Some chemicals will insert themselves into the DNA and interrupt the shape. So an example of this, after some UV light on two thymines that are close together, it's going to make a thymine dimer. And this is going to change the shape of the DNA. Some mutations can alter the bases that are present in the DNA strand and change their base pairing properties. And so some of the mutagens that we are looking at here, so UV, that's a big one, X-rays from medical devices, carcinogens like cigarettes, processed foods have quite a few, and preservatives can be filled with carcinogens. Cosmetics, cleaning products, viruses, and bacteria can all be mutagens. All right, so this is it for Chapter 17. Have a great day.